Now it's my pleasure to introduce today's speakers, Dr. Stephen Dobson and Johanna Ohm. Dr. Dobson's a professor at the University of Kentucky and a member of the Department of Entomology. He's a medical entomologist by training who has been investigating cytoplasmic incompatibility in mosquitoes and the intracellular bacteria Wolbachia that is responsible for it in some insects. In 2010, he decided to put theory into practice and founded Mosquito Mate Incorporated, a company leading the way on developing and implementing the incompatible insect technique, an insect control method that is based on cytoplasmic incompatibility. Dr. Ohm is a scientist at Verily Life Sciences. Verily Life Sciences is a life science research organization within Alphabet Inc. She's working on their Debug project, where they, as they say, and I'll quote, are trying to stop bad mosquitoes by raising and releasing good ones. Dr. Ohm is the technical lead for Debug's mosquito production systems, and it's primarily focused on developing Debug's next generation mosquito larval rearing systems. The work at Verily is very interesting. We're going to really enjoy hearing about it. With that, let me say welcome, Stephen and Joe.
Hello.
Good. Thanks for your willingness to participate today. Really happy to have you, and I'm really excited about hearing about the work and the work that you're collaborating with Verily on. So, Stephen, let me invite you to share your presentation at this time. Let me know when you've got it up, and I'll let you know whether or not you've been successful.
Okay, am I successful?
Yep. You've been successful, and I can hear you. Your visual's good and the audio's good, and the floor is yours. Thanks a lot.
Super. Well, thanks again, Dave and Hector, as well as the FNIH for the invitation and the opportunity to present today. I guess I first want to start with just acknowledging the many collaborators, funding agencies, without which this work would not be possible. My name is Stephen Dobson. As you've just heard, I'm a professor of entomology at the University of Kentucky, and I'm also the CEO of this university spinoff named Mosquito Mate. Now, Dr. Ohm and I have the benefit of building on the previous presentations in the series. So in listening to the prior lectures on irradiated and genetically modified mosquitoes, you already know that these technologies are all built on the ability of a male mosquito and the fact that males don't bite. They don't transmit pathogens that cause human disease. And importantly, they are highly evolved and adapted to seek out and mate with female mosquitoes, which makes them the perfect delivery mechanism. You've also heard from previous lectures that historically, these approaches that are in which male mosquitoes are being released, historically they're based on irradiation of those males. And there's a number of very successful agricultural examples, which we've heard about in previous lectures, so including the screwworm, the pink bollworm. Some of these have been successful on a continental scale. They require these large rearing facilities and then the release of these irradiated insects. And again, highly successful. You've also heard in the previous lecture about the application of this irradiation technology to mosquitoes, and that historically there have been some complications, and specifically that the dose of radiation that's required to fully sterilize some species of these male mosquitoes can also make them sick. And that's kind of illustrated by this cartoon here. As you can imagine, on this continuum going to higher doses of radiation as you move to the right, at some point you actually irradiate the males to where they're dead. And then we can turn down the dose to where they're fully fit. And importantly, we need these males to be fully fit because they have to get out there and compete against wild type males that have not been irradiated or genetically modified or infected with Wolbachia and all these technologies. And so what you heard about last week from Dr. Boyer is some exciting new developments where they're testing a lot of new methods. For example, switching from irradiating pupa to now trying irradiation of adult males. Also things like hypoxia, irradiating under very low dose oxygen levels. So some interesting new methods to basically what we're aiming for is right here by this with this red arrow, a male that's sterile but fully fit and able to get out there and compete, as opposed to one that may be fully sterile but is not fully competitive. So what we've done and what you're going to hear Dr. Ohm and I talk about over the next few minutes is instead of also trying to improve upon radiation technologies, we've actually moved completely away from irradiation as the sterilant. We're not using genetic modification. Here we're focusing on Wolbachia. So this is, here you can see in green is Wolbachia pipientis. It is an obligate intracellular bacterium. So here you can see it inside the insect cells. The insect's nuclei are in blue. The green is the Wolbachia floating around in the cytoplasm of this insect. And it's a maternally inherited bacterium. So here you can see Wolbachia, this cloud of Wolbachia in the pole of the embryo. It's being passed from mothers to both sons and daughters. So that's going to play a role later on. So keep in mind that Wolbachia is maternally inherited. Males are a dead end for Wolbachia infection. And as highlighted over here in this periodical, the Wolbachia is known to occur in, or at least estimated to occur in over half of all insects. It's a very successful bacterium. It's found in bees and butterflies and beetles. And in this high school experiment, basically you can send your students out no matter where you are in the world and collect a random assortment of insects and bring them in and do some PCR or molecular testing, and roughly half of them should be infected with Wolbachia. So it's a tremendous evolutionary success, and part of the reason for that is its ability to manipulate the reproduction of its host. And what we'll be talking about today is called cytoplasmic incompatibility. And you've heard about Wolbachia a couple of lectures ago, and I'm going to spend a few slides just contrasting what we're going to be talking about today is very different from what Dr. Moreira talked about a couple of weeks ago. So yes, we're doing this in some of the same species of Wolbachia, same species of mosquitoes, some of the same species of Wolbachia, but it's a very different approach, and I'll try to highlight that over the next few slides. So in general, the overall application is very similar to what you've been hearing about in past lectures in this symposia. It is the repeated inundative release of these male mosquitoes. So that's what's illustrated by this wavy line. It's pulses of males going into the population. You got time on the x-axis. And then this is an indicator of population size on the y. Here's the wild type population. As the same level of male releases take place, an increasing proportion of the population becomes sterilized or made incompatible. The eggs don't hatch. And here's what happens. So this is the Wolbachia infected male that's being released. When the wild type mates with wild type, you get wild type offspring, as you would expect. But in this example, where the Wolbachia infected male mates with the wild type female, you get this incompatibility. And the result in these mosquitoes is that the chromosomes don't segregate correctly. You get, instead of the chromosomes lining up across the metaphase plate and dividing normally, you get this heterochromatic clump of DNA, and the paternal chromosomes never segregate, and so therefore the mosquito can't survive as a haploid, and it leads to early developmental arrest, and the eggs don't hatch. Now, CI, or cytoplasmic incompatibility, takes a lot of different forms. We could spend the whole 25 minutes talking about examples again in butterflies and bees. There are many different variations. But for the sake of this presentation, I'm going to focus on a couple of examples. This is an example that we call unidirectional incompatibility. And this happens where one of the types, we'll say this is a mosquito type, is infected, shown by green. One is uninfected, shown in white. And in this example, well, and in all the examples that I'm going to show today, the same rule applies. And that is that if the male has a Wolbachia infection that's different from his mate, then it's an incompatible cross. So in this example, here's the male that has the green Wolbachia infection. He's mating with a female who's uninfected. The white means uninfected. And so it's a different infection type, and so therefore it's an incompatible cross. The eggs don't hatch. In all the other crossing variations, you do get egg hatch, and the Wolbachia type is inherited from mom, and so therefore these all carry the infection. And so as you can see, in this scenario, the Wolbachia has an advantage relative to the uninfected females, and this is what leads to the spread of Wolbachia into the population, which I'll show in just a moment. So a second example, and I could say that the previous example, one would be Aedes aegypti. Aedes aegypti mosquitoes are not normally infected, not naturally infected with Wolbachia. And so when you hear us talk about aegypti and Dr. Ohm's presentation later, that's what's going on there. This example might be Aedes albopictus, which is naturally infected. And here what you can see, instead of having uninfected individuals, we have two different types of Wolbachia, a red type and a green type, if you will. And in this case, the same rule applies. If the male has an infection type that's different from his mate, it's an incompatible cross. And so that's true in this case, where the green male mates with the red female, and the reciprocal cross. It's only when both the male and female have the same Wolbachia type that it's a compatible cross, and the eggs hatch, and again, they're inheriting the Wolbachia from their mother. So just to quickly review these two contrasting strategies, a couple of weeks ago from Dr. Moriera, you heard about this population replacement approach. Both of these are based on Wolbachia. Both of these are focused on Aedes, at least include Aedes aegypti. And so this is the Eliminate Dengue, now known as the World Mosquito Program, taking place in Brazil. Importantly, so with the population replacement, you heard a couple of weeks ago, their intent is not to necessarily reduce the population size. They're not using it as a pesticide, but instead they're establishing the Wolbachia type in the field. That's the goal. So with success, you don't have fewer aegypti, they're infected with this Wolbachia type. And the rationale for that is, the thinking is that the mosquitoes that carry the Wolbachia may live a shorter period of time. They may be less able to transmit the dengue virus, for example. So that's population replacement. Importantly, because Wolbachia is only inherited through the female, it's maternally inherited, this approach necessarily requires that you release females. You have to release females for it to succeed. Oops. Sorry. Wolbachia suppression, which is what we're focused on today, and again, contrasting the two, is focused on the release of males. And here the goal is not to replace the population, but to reduce the population size. So very much like an insecticide or a pesticide. And we'll be talking about examples today for both Aedes albopictus and Aedes aegypti. So just to show that in another example, this is a model, kind of maybe a segue to the next series on modeling. But this is showing a population replacement approach, such as some of the CRISPR or gene drive type systems, or the Wolbachia system that we heard about a couple of weeks ago from Dr. Moriera, where the Wolbachia population replacement. So this is an island, if you will, a simulated island. This number here is an indicator of time. And then the y-axis, if you will, is the number of females. And so what you're seeing is at the start, and I'll restart this once it completes. At the start, this island is uniformly a single Wolbachia type. And then at time point 0, there's a seeding of the population, a single introduction of these Wolbachia infected females. And then what you're seeing is, because you release the Wolbachia-infected females, because of this incompatibility and the advantage that it provides to the infected cytotype, you're seeing the green type becoming replaced with the Wolbachia-infected red type. And so, yes, and there's examples. We've heard about this a couple of weeks ago, taking place in Australia and Brazil. So then, just to contrast this with the Wolbachia suppression approach, here, same island, indicator of time, you start with this green population. Here we're releasing these Wolbachia-infected males in the center of this island. And note there's this red area, it's not increasing like it did in the last illustration. And that's because we're only releasing males. And Wolbachia is not transmitted through males. And so we're just causing this incompatibility. We're pushing the population down just like you would with the radiation. And there's no corresponding increase of the introduced Wolbachia type, because Wolbachia is not passed through males. And so with success, by eliminating the mosquitoes from the center of this island, you could then reallocate those males, move out to the periphery of the island, and eliminate the population. So that's the approach that Dave introduced with, say, Dr. Lavin back in the 60s. So with the modeling data, we then moved into doing laboratory cage studies as well as large greenhouse trials. This is some results. And let me just kind of explain what you're looking at here. You've got time on the x-axis. This is in weeks. The y-axis, again, is a number of females, indicator of population size. And we have two parallel greenhouses, two replicate greenhouses. One is in red and the other is in black. And then you've got these gray boxes, and this indicates the release of Wolbachia-infected males. So these Wolbachia-infected males are only being released during this period shown by these gray boxes. And so what you can see in the absence of releases, these two replicate cages are tracking each other fairly well. But then in this second experiment where we release the Wolbachia-infected males only into the red greenhouse, if you will, what you can see is the red line going down and being eliminated within about 8 weeks. Here's another repeat with the same outcome. Again, the black cage, the control cage that's not receiving the Wolbachia-infected males remains stable and persists. So we took this data to the EPA and requested a permit to conduct field trials. And this took place in around 2012. We started talking with the Biopesticides and Pollution Prevention Division. This is what it looked like at the time. We've subsequently been adopted by a new branch of the EPA, Novel Technologies branch. But this is how we started. And after about a year or two of working with them, we received experimental use permits, initially for Aedes albopictus and then subsequently for Aedes aegypti. And so this is the WB1 strain, which you'll also hear about in Dr. Ohm's talk. And then this is the ZAP strain of albopictus. And so Dr. Ohm is going to give us, she's going to do a deep dive into what some of these trials look like. I'm just going to do a very quick overview. As you can imagine, in the initial work, we focused a lot on logistics, some very applied problem solving. So we're based in Lexington, Kentucky. That's where we're doing the rearing of these mosquitoes. And yet some of our field work was taking place out of state. For example, in California, this is work that was done with Consolidated Mosquito Abatement District in Fresno, or Clovis. And so we had to develop approaches to package, to rear, separate, remove the females, and then package these males in such a way that they could be shipped and released in California. And remember, this is what I talked about earlier, the importance of these males being competitive. And so is it possible that all this handling and shipping and who knows what the temperature is as they're going, that maybe by the time they get to California, maybe they're less competitive? And so here's a picture of them being released in the field. And so we spent a lot of time examining the shipped males on the receiving end, looking at things like this cage assay. This should look familiar from last week, looking at male competitiveness. So here we're varying the ratio of incompatible males. Again, this is on the California side after shipment, going from 0% incompatible males to 100% incompatible males, and then measuring the egg hatch here on the y-axis. This line is what one would expect if they are equally competitive. This is just a quick summary showing lab cages as well as some of these larger greenhouse or mesocosm cages. And in short, we were able to determine that the males actually survived shipment very well and remained competitive on the far end. We also were looking at things that are very specific questions of the regulators. So for example, female contamination rates. How often is it that a female happens to be in one of these male release tubes? Looking at things like, could Wolbachia become unintentionally established at the release site? So remember with Dr. Moriera's talk a few weeks ago, that's the goal. They want to establish Wolbachia at the release site. But in contrast, we're releasing only males. Males are dead-end hosts for Wolbachia, and so therefore the Wolbachia should not become established. But doing PCR reactions, we can screen to look for that and actually confirm it. And then obviously looking at things like given a known release rate, if we do PCR assays on the collected males, what proportion of them are the Wolbachia release males? And so we can establish a release ratio from that. So moving forward very quickly, because I'm conscious of the time, this is what a typical trial looked like. So you can see we've moved from California. This is now Stock Island, Florida, which is in Monroe County. It's down in the Florida Keys. You'll see a recurring theme throughout my presentation, but also in Dr. Ohm's talk in a few minutes. There's one or more treated sites, one or more untreated sites. We like to keep a distance between them of at least 1 kilometer just to minimize any released males migrating across and affecting the untreated site. We're doing things like measuring the adult number using biogens or BG traps. Also collecting eggs with ovitraps, looking at both egg number, density of the population, but also egg hatch rates. Because of this incompatibility and because we're releasing millions of these males, we anticipate the hatch rate is going to go down. We love working on islands because that gets away from some of these immigration effects that you see where females from outside the release area are immigrating into the release area and laying eggs that hatch. So it's always a complication. But also doing things like going into, here's an example of Miami, where it's not an island. And in the release and treatment area, there's plenty of infestation and plenty of opportunity for immigration. Again, you're going to hear a lot more about this in Dr. Ohm's presentation. But generally, the idea is we like to pick 2 sites that are very similar demographics. We like to, if possible, have substantial monitoring data prior to starting the male releases just to confirm that they're similar. And again, you'll hear more details about this in Dr. Ohm's talk. So just to wrap things up, a very quick synopsis. This is where we have done trials to date. The pink color is for the Aedes aegypti. It shows both the city where the releases took place as well as the years. The yellow is the Aedes albopictus work. And then down here, this Gantt at the bottom is showing you for albopictus that we progressed from lab studies all the way through EUP permitted field trials to where we now have full registration for the ZAP mosquitoes. Aedes aegypti started behind ZAP a little bit later. We have conducted those field trials. That data is currently under review at the EPA, and we are hopeful to receive a decision from them in 2021, this year. And just to highlight, I'm sure Dr. Ohm is going to do this as well. You can see that a lot of this work has been published. If anybody is interested, if you don't have access, please contact me, and I'm happy to provide additional information. And the one last thing I want to do before I turn things over to last slide, before I turn it over to Dr. Ohm, is to emphasize that while today we're focused on Aedes species, aegypti specifically, and albopictus as well, just as Dave mentioned in his introduction, this technology has the potential to be extended to other mosquito species, anopheles, which are important at transmitting malaria as well as other pathogens. And here's an example where the target's not so much human disease, but a bird pathogen. In Hawaii, the invasive culex are transmitting avian malaria between birds and putting some endangered species at higher risk of extinction. And so happy to provide this additional information if someone's interested. So with that, I should stop. I think I'm over my time, and I will yield the floor to Dr. Ohm.
Hi, thanks, Stephen, for that great overview of how we can use Wolbachia-infected males to effectively suppress wild populations of mosquitoes that transmit disease. Let me just pull up my slides here. So the Debug project partners with Mosquito Mate and government agencies to try to make this Wolbachia-based mosquito control approach scalable, with the hope that if we can make Wolbachia-based mosquito control really widely available, we could have a big impact on the diseases that these types of mosquitoes transmit. So as David mentioned earlier, Debug is a project that's based at Verily Life Sciences. And we develop tools, including hardware and software tools, to really make this process of producing massive numbers of Wolbachia-infected male mosquitoes more efficient. As Stephen really thoroughly went over, Wolbachia is a bacteria that naturally occurs in most insect species. And we use a trans-infected line of Aedes aegypti that has Wolbachia infection, which when released into wild populations, has this phenotype called Cytoplasmic Incompatibility, which means that our released Wolbachia-infected males are incompatible with the wild females that are not infected. So as Stephen also showed in his little video, what happens is over time with enough introduction of our Wolbachia-infected males, those matings are incompatible, the eggs are unable to hatch, and over time we can really rapidly drop the population of wild mosquitoes. For this technology to be effective, we need to have consistent large numbers of Wolbachia-infected males released into the wild so we can outcompete the wild males and ensure that those wild females get mated with the Wolbachia-infected males. The eggs won't hatch. Over time, that population gets reduced. The one tricky part about producing a lot of Wolbachia-infected males is that mosquitoes have a very complicated life cycle. I think most of you listening probably have some familiarity with rearing mosquitoes, or maybe have even produced mosquitoes in a large-scale production facility. Some of the complicated aspects of their life cycle is that adult females require a blood meal in order to produce eggs. This means that we have to have a constant supply of fresh blood, and we also need to have a cold supply chain in order to get that diet for the adult females. The other really interesting thing about rearing mosquitoes is that unlike a lot of insects that are mass reared for sterile insect technique, mosquitoes have both an aquatic stage, so all the juvenile forms develop in water, and the adult stage is terrestrial. So when we want to mass produce mosquitoes in a factory, we have to accommodate both the aquatic juvenile stages and the terrestrial environment that the adults need. And also, as Stephen mentioned, when we use the Wolbachia-based approach, we need to really make sure that we only release male mosquitoes. Female mosquitoes that are infected with Wolbachia are compatible with our Wolbachia-infected males. And so if we want to make sure that we suppress the population, rather than replacing the population, we need to do everything we can to make sure that we only release males, avoid releasing females, and cause suppression rather than replacement. The last thing that makes mosquito rearing and releasing difficult is there's a limited dispersal ability of mosquitoes in the field. So mosquitoes can only fly so far. if we want to suppress a wild population that is broadly distributed, we need to make sure that we distribute our Wolbachia-infected males really evenly across our release sites so that they have a fighting chance to go out, find those females, and effectively suppress the population. On Debug, we work on a number of different types of tools to try to make this process more efficient. I'll talk about 3 of our main tools today and just give you a brief overview here before diving into what these tools look like. We've developed an automated larval rearing system, which collects larvae from recently hatched eggs and then inputs those earliest stages of mosquito larvae. So we call that the L1 stage, when they're freshly hatched, very tiny larvae, and it rears them through until they pupate. So we have an automated system that requires very little human attention to rear those mosquitoes all through their aquatic juvenile development. After pupation, we've developed a number of different tools to sex sort the mosquitoes. So again, we want to minimize the chance that we'd ever accidentally release a female with our males. So we have 2 types of processes, one that mechanically separates pupa at the pupal stage and separates males and females based on differences in body size. And then we also have an adult sex separation machine that I'll show you how it works based on an industrial vision system. Finally, we've also developed tools to make releases more efficient. So we have release now in a number of different types of environment, and we've developed customized tools and software to make these releases a bit easier depending on the environment we're releasing in. So to start with our mosquito rearing system, in this picture, you can see what really typical mosquito mass rearing looks like. There's lots of trays, lots of shelves filled with larvae and mosquito water and food. We have a human here that has to go in and check on the larvae, monitor their development, and maybe once or twice daily deliver food to these larvae. What we thought here at Debug might make this process a little bit easier is if we could remove the human needed in this process. So we've developed an automated larval rearing system, which also looks very similar to the manual setup. So we have lots of trays filled with water and lots of larvae. And unlike the typical picture of manual mass rearing, we keep these trays of larvae inside an enclosed container that regulates the temperature really tightly. So the difference between any of our shelves in our automated larval rearing system is within 0.5 degree Fahrenheit. So really, really tight temperature regulation that took a lot of engineering to create. And that means that we can have a really tight development window on how long these mosquitoes take to develop. The other thing that's really nice about this system is that we've saved a ton of manual labor. I'll zoom in and show you what this system looks like. So on the first day of larval rearing, we use a laser counter to precisely count mosquito larvae after they've hatched. So these tiny L1 larvae get passed through our laser counter, precisely counted into a dose of larvae that we then put in these plastic bags. So the plastic bags you can see on the left are actually the same exact material that's typically used for packaging hot dogs or other type of foods. And we add precisely a measured amount of water that's also automated in its add. And then we also add an exact amount of food. So once those bags of food, water, and larvae get packaged, they get passed by our storage and retrieval robot into the interior of our larval rearing system. So the larval rearing system has a robot that runs down the center aisle and can store and retrieve these bags of larvae on index shelves that make it really easy for then each day of the rearing cycle. The software allows us to schedule what time we want these mosquitoes to be fed, and without the need for a human to go in and manually do those feeds. The other thing that's really nice about this system is using these plastic bags means that day 7, when our development process is ready to collect the pupa, the bags can get disposed. So we don't have to clean a bunch of trays. We save a lot of labor in terms of cleaning as well. So after the mosquitoes have developed into the pupal stage, this same robot will then pass the mosquitoes to our sex sorting machines. So that's what our larval rearing system looks like. We first used this rearing system for our first field trial, which took place in Fresno, California in 2017 and 2018. And that was our first test of using this system. And we had a number of questions as to whether we could use an automated larval rearing system to replace manual rearing and still be able to produce consistent yields of male mosquitoes that were of consistent quality. So again, the goal here was produce as many mosquitoes as possible and make them as healthy and consistent in quality as we can. So this is showing data from 2018. So the insects that we produced for our Fresno field trial. And what we measured here in the data shown in the graph on the upper left-hand side of my screen is the average adult number of males that we got out of our larval rearing system and passed through our sex sorting process compared to the number of L1 larvae, so those early stage larvae that we input to the system. What you can see is obviously there's a little bit of noise in the data, so most biological processes can be a little bit bouncy, but we were excited to see that the average here was about 70%. So of the 200 plus batches of mosquitoes that we reared over the course of 2018, each one of those batches had anywhere from 50 of those small plastic bags that I showed in the previous slide to several hundred. And we consistently were able to get a majority of those larvae reared into pupa and then released as adult males into the field. With that 70% average yield, we estimated that our automated larval rearing system could produce upwards of 2.5 million male pupa per week, which we took as a big success for validating that automated larval rearing systems can indeed replace the human labor of normal rearing processes to consistently produce lots and lots of mosquitoes. The other thing that we do as a check that our males are consistent in size, and we use this as a proxy for male quality, is measuring the length of our males as adults. So the idea is that consistent size and relatively large males tend to be healthier. And if we monitor their size, we can get an indicator of how consistent the rearing process is. So again, this is the same mosquitoes that we used in the previous graph. This is data showing every batch that we reared over the 2018 field season and the sizes in micrometers on the y-axis. One thing that I think is really impressive about these data is we had 14.4 million male mosquitoes released into the field during that 2018 field trial. And the average of all of those body lengths from millions and millions of mosquitoes created a standard deviation of less than 1 millimeter in size. So really consistent sized mosquitoes, demonstrating that this system could definitely be used for consistent production of high yields and consistent quality. So again, this data is from 2018. And since 2018, we've made a number of changes to our larval rearing system. We're still experimenting with the optimal protocols used in this system. But just to give you a taste of some of the things we've been working on more recently, is we think we can make the automated larval rearing system more efficient with improved diets and possibly different density protocols. So the other way that we estimate male quality before releasing is by running longevity assays and evaluating how long do these males live after we release them in the field. So on the right, you can see a graph of the days after we would have released males, and on the y-axis, the proportion of males in those longevity cages that are still alive. In 2018, we used a pretty standard mosquito diet. So commonly used is bovine liver powder. We used that same diet throughout the 2018 field trial and saw that our male longevity was about 4 days after release. We've done a number of experiments trying out different diets and found that with changing the diet, we can actually extend that lifespan by about 1.5 days, which is a huge gain for us because it means that the mosquitoes can hopefully live longer in the field, have more time to compete with wild males, and have more matings with the wild females. The other thing that was exciting about using an improved diet was that it seems like with a healthier diet, we could also bump up the density in our larval rearing system. So in 2018, we used a relatively low density rearing protocol. And what I'm showing in the upper left here is that the male longevity on that diet is increased with our new standard diet. So these mosquitoes are living close to 6 days on our improved diet protocols. But when we use that same diet and increase the density, we still get longer longevity than the low density protocols that were fed with bovine liver powder. So really promising data that suggests we can get the efficiency of the automated larval rearing system even higher if we change the densities that we're using and the diet that we're using. The next area of technological development that we've been working on is the sex sorting process. So again, it's really important that for a Wolbachia-based suppression approach, we release only male mosquitoes rather than females. Accidental release of a female could result in establishment and population replacement. The most common way to sex separate Aedes aegypti is through a machine called the Hox machine, which is shown in this picture. So you can see that there's two kind of clear bands of pupa in this picture. The female pupa are bigger than male pupa. And so you can size separate with the Hox machine by having the larger female pupa get stuck in the wider section of the space between the two plates, and then smaller males fall through the narrower slot on the bottom. This machine works really great. It's very effective at separating male and female pupa. But one thing that Debug, again, wanted to see if we could do is eliminate the need for a skilled human operator. So we started developing a sex sorting process that eliminates the need for that human operator. And it starts with an automated sieve to similarly separate male and female pupa, but done by robot instead of by human. So on the left here, we have the size distributions of male and female pupa. These measurements are taken at our South San Francisco rearing facility. You can see males tend to be smaller, females tend to be bigger, and we can design a sieve to separate the males and females. The females, again, are bigger, so just like the Hox machine, they get stuck behind, and the males pass through our sieve system. We, again, collected data on how efficiently we separated males from females throughout our 2018 field season in Fresno. So again, the x-axis here is the batch number from that 2018 field trial. And then on the y-axis, we are reporting the fraction of male mosquitoes that are in our pupa samples on the other end of the sieve. So really encouragingly, the vast majority of female mosquitoes don't pass through the sieve. So we have about 94% of the pupa after sieving are male, which is great, really pretty high efficiency, but that means that we still have a 1 in 40 chance that any one of those pupa could be a female. So to further reduce the risk of potential female contamination, we then take these sieved pupa and pass them to the next stage of our sex separation process. So we do this at the adult stage. Adult mosquitoes have really distinct morphological differences between the sexes. So we can use an industrial vision system to separate male and females in the adult stage based on these characteristics. On the left-hand side, you can see a picture of what adult Aedes aegypti female looks like. She has a very fine antenna and a relatively wide abdomen, which is very different morphologically from what the males look like. On the right here, you can see a male mosquito. He has really bushy antenna. Some people equate this to a mustache kind of look on their antenna. They also have claspers at the base of their abdomen, and their abdomen's a lot narrower. So using these distinguishing characteristics between males and females, our industrial vision software captures a picture of every single adult male that we rear and that has gone through our pupa sieve. As those males pass under the industrial vision system, they get their picture taken. The picture then gets evaluated as to whether that's a possible female contaminating our males, or it labels it as a male. What this looks like in real time is we have our highly trained mosquitoes walking single file down these sex sorting machine lanes. And we have a little trap door in the center of our lane that shuts behind each individual mosquito. After the mosquito passes through the door, it gets its picture taken. Our industrial vision software then decides whether that image is of a male mosquito or a female mosquito. The males get accepted into our release containers, and the females go into a reject path that removes them from the process. So importantly, this reduces our risk of female contamination by a number of orders of magnitude, and really reduces the risk that we could potentially release a female. Because we wanted to make sure that our industrial vision system couldn't also be creating the risk of a female contaminant, we add another layer on top of that that passes the images that our camera takes to a machine learning classifier. The machine learning algorithm then ranks all the photos that the industrial vision system takes and classifies them as how male-like or female-like those pictures that were originally labeled as male are. Any pictures that machine learning catches as potentially female-like in the male labels then get passed to a team of expert reviewers to manually do a double check that those images are indeed of male mosquitoes and not female mosquitoes. So to give you an overview of what that multi-step process in our sex sorting path looks like, again, it starts with pupa that are a mix of males and females. We pass them through our pupa sieve, which mechanically separates the larger females from the smaller males. That leaves us again with about a 1 in 40 chance that we have a female contaminant in our males. And then we do this multi-step process on the adults. So we start with our industrial vision, taking pictures of the mosquitoes, labeling them male or female dependent on what those pictures look like. And that reduces the chance of a female to about 1 in 40,000. When we add on top of that our machine learning algorithm and the expert review step, combined, we get a great risk reduction. So we estimate that if each step in this process is independent, at the end, we have less than a 1 in 840 million chance of ever contaminating our males with a female. So this is in comparison to just mechanical separation at the pupal stage, 1 in 40 compared to 1 in 840 million. So a big improvement from mechanical separation alone, and something we're really excited about in terms of reducing the risk of causing establishment. The final area that we've been working on developing custom tools to release mosquitoes in our field sites is in release devices. Again, our first field trial took place in Fresno, California, which looks like this video in the lower left-hand side of my screen. You can see that the houses are typically low to the ground, like 1 or 2 story homes. The streets are really nice and wide. Most mosquito habitat is really accessible by car. So for these types of sites, we developed a release van that releases mosquitoes out of a port on the side of the van. The drivers of these vans get software that maps all the release sites in our release areas that we want them to drive by. So as a driver of one of these release vans, you you get to play this fun game of Pac-Man, where you're driving along the streets, and as you pass or collect one of our release site points, the van gets automatically triggered to release mosquitoes out of the port on the side of the van. And the software collects data on where those were released when and the numbers of mosquitoes that were released at each point. So that worked really well for our Fresno field trials, but since Fresno, we've expanded into other areas that are really different in terms of the cityscape and landscape that we have to release into. On the right, you can see a picture of our Singapore release site in Campinas. And you can see that the buildings are really different than a lot of the buildings in Fresno. So we have tall, high-rise apartment buildings with lots of outdoor balconies that might have plants or tiny pots that mosquitoes love to breed in. So for sites like this, we've developed alternative technologies. Instead of releasing on the ground, we're also working on containers that can be used for manual releases, spreading these mosquitoes to places where they might be hiding on upper level stories. So all of those tools and technologies we've developed to make production and releasing easier are only valuable if they can also effectively lead to suppression of wild populations of mosquitoes. So to that end, we've conducted a number of entomological field trials to look at whether our Wolbachia-infected males actually reduce wild populations of biting females. So we've conducted a number of field trials. Again, we started in Fresno here in California in 2017, and we have since expanded to conduct releases in Singapore, which is still ongoing work. We're expanding and continuing releases through this year. We've also had releases in Innisfail in Australia back starting in the fall of 2017. And most recently, we've been working in Ponce, Puerto Rico, where we're partnering with the US CDC mosquito mate and the Puerto Rico Vector Control Unit to do an entomological field trial there as well. I'll start by giving you an overview of our results from Debug Fresno, which again was our first field trial. We started in 2017 and continued throughout the field season in 2018. On the left is a map of our treatment and control sites. We selected 3 treatment sites shown in orange that are labeled as T1, T2, and T3. And then we had 3 geographically matched control sites, C1, C2, C3. As Stephen mentioned, we carefully selected these sites to have controls and treatments that were matched geographically and similar in the number of households and similar in the mosquito populations. Zooming in on this control area 1 and treatment area 1, you can see that in all of our treatment and control sites, we also had pretty extensive trapping. So in black, you can see where we placed our BG sentinel traps, which collect wild females. And then in gray, we have ovitraps. So a combination of these 2 types of traps gives us 2 different types of data. The black dots that collect the number of females gives us an indication of how well we're suppressing the wild population. So when we compare the numbers of females in traps from the BG sentinel traps in our control areas to our treatment areas, the hope is that if our treatment is working, we'll see much fewer female mosquitoes in our treatment sites than in the controls. Some of our treatment sites were close to the control area, so T1 and C1 were close together, so we also had this buffer zone in between them where we trapped but did not release mosquitoes. The one thing to note about our treatment site 2 is we did have a substantial risk and concern that we would have migration from mosquitoes outside the treatment zone immigrating in. So we also had a buffer treatment there that was excluded from analysis, but still had releases take place. As I mentioned earlier, this was the first time that we used our automated larval rearing system to conduct a field trial. So The first success of Debug Fresno was that we were able to use this automated system in combination with our sex sorting machines and new release fans to successfully deliver male mosquitoes into the field. This graph on the bottom here shows you the numbers of mosquitoes that we were releasing every day of the field trial in 2018. The y-axis is the number of male mosquitoes that we released times 1,000. And then shown in the different colors is a stacked sum plot of the numbers of mosquitoes that went into each of the 3 treatment sites. The numbers vary between the treatment sites depending on how many households there were and what the local mosquito population looked like in each of those sites. So as you can see, we had consistent production of mosquitoes. We were releasing close to 550,000 per week in Fresno, covering about 3,000 different households. And these gaps you can see on Memorial Day, 4th of July, and Labor Day are the US holidays. So we give the mosquitoes a day off. The second thing we were looking at in Fresno was to see how effectively our released Wolbachia mosquitoes were at suppressing the wild population of Aedes aegypti. So Fresno is a desert-like area in California, and it has strong seasonality in the mosquito population. So this is a graph of what the mosquito population looks like across season in our control sites. So pretty typical of places with strong seasonality in the mosquito population. You can see in early spring, there's not many mosquitoes around. And as the temperature increases and the climate becomes more favorable for mosquitoes to develop, you see a rapid rise in the mosquito population size. By mid to late summer and early fall, you have a really strong peak in the mosquito population numbers. So lots and lots of mosquitoes getting caught in our traps. And then as it cools down again and we go into fall and winter, the population drops again, and we see very few females in those traps. We started releases in early April, shown in this first dashed line, and then we ended releases in mid-October on the second dashed line. What we were expecting, if our suppression approach was working, was that in our treatment sites relative to controls, we wouldn't see this strong peak of increase in mosquito numbers. So ideally, in our treatment sites, we would see a flat line and 0 or very few female mosquitoes getting caught in those BG traps. And what we saw was really encouraging. We saw an average of over 95% suppression between our treatment sites and controls. So again, this red line is our treatment areas averaged across those 3 treatment sites. And what you can see is during that peak, when we see tons and tons of mosquitoes in our treatment sites, or in the control sites, our treatment sites barely blip above 0. So really strong suppression. The biggest effect we saw was in the most isolated treatment site, and we saw upwards of 97% suppression in that site, but an average of 95 was really encouraging. So since Debug Fresno, we've expanded into other treatment sites, but I'll go through those in just a couple of slides here. And if you'd like to read more about our Fresno-based field trial, we published these results last year in Nature Biotechnology. And you can read about some of the automated tools in more details and the results from Debug Fresno. So as I mentioned, we now release in Singapore. Singapore is a very different environment than Fresno. It's a tropical environment. There's much less seasonality in the mosquito population throughout the year. And we are dealing with lots of high-rise buildings, mosquito habitat that's dispersed across the vertical landscape as well as the geographic ground level landscape. We started releases in the fall of 2018 in an area called Tampines, which is in eastern Singapore. Our first release site was this small area outlined in yellow that we called TP01, and we've since been expanding to bigger sectors throughout Tampines. So one key difference with Singapore is is that the climate is really different, as I mentioned, from Fresno. And also we partner with the National Environment Agency to rear the mosquitoes locally in Singapore instead of using our San Francisco factory. But just like Fresno, we saw really encouraging results so far in Singapore. So these releases are still ongoing, but in partnership with the National Environment Agency there, or NEA, we've collected data from the gravitraps, which also collect adult females, and seen that over time in areas that we've released Wolbachia-infected male mosquitoes, we see suppression of the wild population of biting females. So compared to sites without releases, this graph is sort of a cartoon version of the data that NEA released on their Facebook page. And in orange, this orange line shows that sites without releases, so our kind of control areas where we're not treating with Wolbachia-infected mosquitoes, we see larger numbers of potentially infectious mosquitoes in those areas. In areas where we've consistently done Wolbachia releases, we see much fewer mosquitoes. And unlike Fresno, Singapore also has widespread dengue. So really encouraging for us was that NEA has also seen really substantial decreases in dengue cases in the areas that we've released mosquitoes. They estimated between a 65% and 80% reduction in dengue cases in our Wolbachia treated areas. So really exciting in terms of not only do we suppress the local population of mosquitoes, but that also translates into reduction in diseases like dengue. As I mentioned, we started early in 2018 with a small release site, which is outlined in this black cartoon in 2018. And we've since been expanding to other areas in the region of Campinas. And every place that we've started expanding into, we've seen corresponding decreases in dengue cases. So we'll continue this work throughout this year and hope to see this pattern of reduced mosquito populations and also reduced risk of dengue. Finally, our most recent field trial that we've started is Project COPA that we've done in partnership with Mosquito Mate, Puerto Rico Vector Control Unit, and the CDC. This trial takes place in Ponce, Puerto Rico, which is the one of the bigger cities in Puerto Rico. Puerto Rico has the highest transmission of mosquito-borne diseases anywhere in the US. So it's a great place to conduct a Wolbachia-based suppression approach to not only monitor entomological impact on releasing Wolbachia-infected males, but here with the CDC, we're also conducting an epidemiological trial. So this trial just started in mid-2020, but over the next year, we'll see both hopefully a suppression in the local population of mosquitoes, and we're also looking for epidemiological impact in reducing the incidence of disease. One thing that's really unique about our Puerto Rico trial that is unlike either our Singapore trial or our Fresno trial is that we don't have a mosquito factory locally in Puerto Rico. In Singapore, I mentioned we rear mosquitoes in partnership with NEA at their mosquito factory in Singapore. In Fresno, we reared our mosquitoes in South San Francisco and could easily drive the mosquitoes to our release sites. In Ponce, we actually pack and ship mosquitoes all the way from our San Francisco facility, ship them to Ponce where they get delivered and released. So it's a distance of about 3,600 miles, and we hope that those males arrive happy and healthy and can go out and do their job in suppressing the wild population of mosquitoes there. We don't yet have data on our suppression numbers or the epidemiological impact. We're blinded from the epidemiological results until the trial is over. But we do have some promising data that our packing and shipping methods, which we newly developed just for this field trial, are working hopefully as intended. So again, male longevity is a good indicator of the quality of our males, and should hopefully correlate with their ability to be competitive in the field. So we've looked at both survival of mosquitoes that we've reared in San Francisco and held back and kept in our facility here, as well as mosquitoes that we've packed and shipped, go through this process, arrive 3,600 miles away, get unpacked and released into Ponce. So we've done these sample size numbers are the number of packages we've either shipped or not shipped, not the number of mosquitoes. We've shipped hundreds of thousands of mosquitoes for these longevity assays. And what we've seen really encouragingly is that the average lifespan for mosquitoes that have been held back or packed and shipped is actually identical. So we're still getting over 5 days of adult male longevity after shipping, whether they've been held back as controls or shipped to Puerto Rico. So we hope that this data indicates that our males are healthy and happy and doing a good job of outcompeting the wild males in Puerto Rico. Stay tuned for more data from this trial. We'll hopefully soon have data on suppression and epidemiological impact. With that, I'd like to just thank everyone that's worked on this project with me at Debug, and say that if you're interested in getting involved in working at a mosquito production facility, we're hiring. You can check out Verily's website at verily.com/roles. And I'd also like to thank Stephen, Dr. Dobson for co-presenting with me, David and Hector for organizing this series, and I'd be happy to take any questions.
Great. Thanks a lot, Joe, and thanks a lot, Stephen. We do have some questions, and I think I'm just going to jump right in and start them. Stephen, I think this first one is really pretty much for you. And the question is this: if 50% of all wild-caught insects are infected with Wolbachia, what are the chances that 100% of all the wild female mosquitoes of the target population will not be infected with the relevant Wolbachia strain prior to your intervention? And I guess it goes on here. Let me just continue on here. There's a comment. It would seem that even if a tiny minority of wild females were infected, a Wolbachia suppression method would grant those minority infected females selective advantage and would eventually, thwart the suppression. So let me let you respond to that. And there's a second part of this question, which I'll get to after you respond to the first part. So go ahead.
Yeah, and you can hear me?
Yep, can hear you.
Yeah, it's a great question, and I'll start with an apology because I may have confused the issue. When I talk about 50% of insects being infected, what I'm referring to is not so much that half of one species is infected. It's just that, like Aedes aegypti does not have Wolbachia. There's been some spotted reports but we looked very hard and been unable to find a naturally infected Aedes aegypti individual. So that's one clarification. Whereas albopictus, they're pretty much, we can't find an uninfected albopictus. The second part is a really, really important point. And I'm often asked, like when Dave, when he was introducing, he talked about the study done by Hans Laven back in the 60s. I'm often asked, well, that was a terrific success. Why did it stop there? Why didn't they continue it at that point? And the reason is that they were limited to working with natural infection types. Laven was able to find a German Culex mosquito that was incompatible with the one that was in Burma at the time. And so that's why it worked. The real breakthrough that happened that allowed projects like what we're doing in Puerto Rico and Singapore and Clovis, California, is the ability to artificially move Wolbachia around. And there are literally thousands and thousands of different strains. And in this case, we moved Wolbachia from Culex pipiens, which is a mosquito, the common house mosquito, into albopictus. And so the Aedes albopictus mosquito. So that's kind of a long answer, but hopefully gets at some of the important points that you raised.
Yep. Yeah. So I guess the point, the first point was that when you say 50%, you're really talking about 50% at the species level being infected, not at the individual level.
Correct.
Yeah. Great. Okay. Good. I think you addressed the second part of the question, which was really, how many different incompatibility types are there, and what you're saying is that there's thousands of incompatibility types. Okay, great. Joe, there's a number of questions that come from people's fascination with your technology. And this question starts out by saying, with the comment, amazing larval rearing. How do you add food to the plastic bags? Are they open? So they look like they were sealed bags. And so maybe you could comment on that.
Yeah, I can quickly comment on that. So yeah, the bags are sealed on the edges, but they have small holes in the top so that we can drop small amounts of food in, and that's done by the robot.
Yeah, I guess that would apply to oxygen as well, right? I mean, you'd have to keep these things aerated to a certain extent. Yeah.
Yeah, so the small holes that work as feeding holes also allow some air exchange. They're mostly covered by plastic, but there are a couple small holes to let food and air pass in.
When that robot was picking up those bags, were those holes present at the time, or were they put in later?
Yeah. So the holes are pretty small, and allow that, make sure that mosquitoes don't fall out and water doesn't splash out, but that food and air can go in.
Yeah, good. So here's a question about cost. What would it cost for the automatic production of 1 million males? So there's, I guess, the general gist of this question has to do with cost of production.
Yeah, so costs of production obviously will come down as we improve the efficiency of this technology and spread to more places. We don't disclose our costs right now, but with enough scaling of this tool, I think we can be competitive with other types of mosquito control technologies. Yeah.
Okay. I'm going to keep going here with you, Joe, because there's a number of these technological questions. This next one has to do with, do the adult males live long enough inside your factory to allow them to adapt to the factory's environment compared to the wild?
Yeah, so I think this question was also brought up in a previous webinar series, where there's always lab adaptation. Whenever you take wild mosquitoes and bring them into the lab, evolution goes on and you can't prevent some degree of adaptation to the lab environment. So we have colonies that we maintain here. Some adaptation happens, and we do our best to run assays to make sure that they stay competitive with wild males despite those changes that happen from adaptation.
Yeah, just to follow on with that, is there an effort to reintroduce genetic diversity or wild type genotypes into your standing lab populations over time?
Yeah, so we definitely backcross our colonies to wild mosquitoes to make sure that we do have mosquitoes that are closely matched with our release sites. So we have our lines of mosquitoes that are genetically as close as possible to places where we release. And we start with wild mosquitoes that we then backcross to increase the genetic diversity.
Yeah, another question from one of our somebody in the audience was, how many males per minute can your real-time sorter separate? And what's the temperature in the tunnel? And how do you get them? I'll just add this part. How do you get them to walk down that tunnel?
Yeah, lots of questions, all really good questions. I don't think we've ever calculated what the rate is per hour. That's a really good question. Yeah, and the rate varies. So I think when you watch mosquitoes develop, I'm sure lots of people listening have spent way too much time looking at mosquitoes and watched them throughout the different phases of their development. And like all phases of development, there's a ramp up, a peak, and then a decline. So that goes for, the pupation trend. You'll have, the early pupators and the late pupators, and it looks like a bell curve in the pupation timing. And the same is true for the closing. So there's not really a good way to speed up mosquitoes other than temperature manipulation to get them out of the pupal stage and into the adult stage. So we kind of work with their natural eclosion curves. And when they're ready to eclose, they walk down the lanes, get their picture taken, and go into our containers for release. I kind of wish most of the time that we could make that more synchronous and get mosquitoes to go through the lanes faster, but it is a bit dependent on just what their eclosion curves look like naturally.
Yep.
There's lots of parts of that question, so if I missed one of the questions there, let me know.
Yeah, yeah. No, you really gotta, you really have to know your bug here, don't you? So, do you think, here's another question from the audience. First of all, they thank you for your great presentations. And do you think, or have you simulated that in a species that do not present sexual dimorphism, you could achieve higher percentage of sex sorting just relying on photographing adults?
I would think it would be really difficult to sex separate any kind of organism that shows no sexual dimorphism. There has to be something that the vision software can detect to classify it as male or female. So luckily, the species we work with is Aedes aegypti, and as I showed you in the pictures, they have really, really clear differences between males and females. I can't actually think of an example of an insect that doesn't have some type of sexual dimorphism. So I think it would work for most species. I think you'd be a little out of luck if you can't have a distinguishing characteristic between your males and females to separate them.
Yeah. Okay. Here's a broader question. What are some of the, both of you actually, Stephen and Joe, what are some best practices that you've seen for designing these sterile insect release type studies to test the effectiveness of releasing males and suppressing field populations? I guess that could span, a lot of things in terms of best practices, but anything that you could add would be great.
I'll let you start out, Stephen.
All right, sure. Well, I mean, the key thing that I would emphasize that was really a learning curve for me is to just go into this understanding that this is not a petri plate. This is not a lab type situation. Events are going to happen that throw your best design out the window, and you have to adapt. So that's been a recurring theme throughout a lot of our studies. The second thing is just the importance of community engagement. This is unusual. We're going into people's backyards and properties and releasing mosquitoes. And, initially people's eyes get very large when you start talking about that until you explain that they're male mosquitoes and they don't bite. But then the last thing would be just the importance of these abatement districts. I mean, they have spent years developing trust in their communities, and the ability to partner with these groups is really what's made this possible.
Mm-hmm. Before you jump in there, I just wanted to ask you a little bit more about that public acceptance that you've seen, particularly in your Florida work, because there's other new technologies, genetic control technologies, being planned for field trials down in that neck of the United States that aren't getting such warm receptions in some cases. And I was wondering, what kind of reception have you gotten, and what do you attribute perhaps the difference to in the receptions that your technology has gotten? Yeah, go ahead, Steve.
Oh, okay. I wasn't sure who you were asking.
Yeah, that was your from your experiences down in Miami and the Keys,
yeah.
And comparing that to some of the other things going on down there with regard to genetic control.
Well, I can't really, contrast and compare so much. I mean, I'm more familiar with, say, what Luciano was talking about a couple of weeks ago, the Wolbachia studies where they're releasing female mosquitoes. Obviously, that's a bit more difficult sell to release mosquitoes that actually have the potential to bite people. But I can't say with our work in the Florida Keys, again, we relied absolutely on the abatement districts. They organized radio announcements. We had meetings where the community was invited to come in and ask questions. For better or worse, what we experienced was just not a whole lot of interest. We would hold these meetings and some reporters would show up, and not too many interested community members. So, on one hand, it'd be great to have people to interact with, but on the other hand, if it's nice to not be concerning folks too much.
Yeah. So you found that they're fairly accepting of this technology?
That's been our experience, and,
Yeah.
and I really think that this is important for the field on a larger sense, because there's a couple of weird things that are happening with these technologies. One is you're releasing mosquitoes. That's weird. But the second is the type of mosquito that you're releasing, and some people may object to one versus the other. But at least what I think that what Joe and what we are doing, and the others that Costas and Dr. Boyer that you've heard from, that we are addressing one of those 2 points. We're releasing mosquitoes, and hopefully that will become more mainstream and part of the norm.
Yeah. Okay. Joe, I'm going to move on to some, actually, there was a clarification of the question about the sexual dimorphism, and it really had to do with if there was sexual dimorphism at the pupal stage, would you be able to separate them at that stage rather than adults?
Yeah, so there is sexual dimorphism at the pupal stage.
Yep.
If you look at them under the scope, you can see the sexual differences at the base of the pupa tail. It's just really difficult to manipulate pupa. They're in water. To do it in a high throughput way, I think would be a little bit more difficult, even though you can see sexually different characteristics in the pupal stage. So we focused on adults just because we thought it was a little bit easier.
Yeah. Okay. Stephen, I'm going to go back to you about this, because this question sort of relates to what we were just talking about. How long do you think, and this question states this, how long do you think releases can occur before you may need to go back to the lab to find a new incompatible strain? And the questioner says that they were just thinking along the lines of there's a potential that a female could be released and then pass on the incompatible Wolbachia strain, and that could spread to the wild population over time. And you've talked about that, but they mentioned that they get a lot of these types of questions from the public when talking about these potential use of this technology. So
Right.
Yeah. Mm-hmm.
These are great questions. The thing that gives me the most encouragement is what Luciana was talking about a couple of weeks ago, where like with the World Mosquito Program, they are intentionally trying to establish Wolbachia infections in the field. So in our case, we're trying to avoid it. You heard from Johanna that they're at 1 in 800 million. Was that what it was? It's a very small release rate, as opposed to the World Mosquito Program is trying, they're intentionally releasing lots of females, and still in some cases, it's quite difficult to establish the Wolbachia. So I think that those two things, when added together, reduce, or at least mitigate the risk of accidental establishment. And then the second thing that I would say is, with any of these approaches, evolution has to be in the forefront of our mind. And will they potentially, could something evolve that breaks the system down? But in the case where you've eliminated the population, there is no longer room for evolution in those cases. So I'll stop with that.
Yep. Great. Well, we are just running out of time now, and I want to thank Stephen and Joe for really interesting presentation and stimulating and really inspirational, really, in terms of looking for solutions to some of these major technological problems that you're facing in order to make this, fairly, I'll say straightforward, although we know it's not straightforward cytoplasmic incompatibility phenomena, a very useful tool for controlling mosquitoes. So anyway, thanks again for your great presentations, and thanks for participating in the webinar series. Before we sign off today, I wanted to let the audience know that we won't have a webinar next week on March 3rd. But we are starting, as I mentioned in my introduction, a new series starting beginning on March 10th. And this series will be a series of four weeks where we cover ecological modeling in risk assessment of gene drive technologies. And the first presentation on March 10th will be the use of models in environmental risk assessment for gene drive insects, given by John Mumford and at Imperial College and Michael Bonsall from Oxford University. So with that, let me thank the speakers again, and let me thank Hector for my co-host, and thank Tara and Mira on the FNH events staff for supporting this series and with their technical help. Much appreciated. And thank you for the audience for attending, and we hope to see you back on March 10th. Thank you all very much, and have a nice day.
Thanks all.